Thank you, and good morning, everyone. My name is Marta Montero, and I would like to talk to you about our research on the three-dimensional synaptic organization of stratum lacunoso moleculare in the CA1 field in Alzheimer's disease. So, by hippocampal formation, we refer to a set of brain structures located in the medial temporal lobe. Specifically, the hippocampal formation includes the dentite gyrus, the cornu monic fields, and the subicular complex. The present work is focused on the study of the CA1 field. The CA1 field is commonly subdivided into 5 strata. Pyramidal cells, which are the main cells of this region, are located in stratum pyramidale. The apical dendrites of these pyramidal cells extend through the whole thickness of the region, with the most distal parts being located in stratum lacunoso moleculare. Regarding its connectivity, the CA1 field receives and integrates a massive amount of information from different cortical and subcortical regions, sending projections back. However, there are 2 main sources of projections to the CA1 field, which arrive in a layer-dependent manner. Specifically, entorhinal cortex, direct projections from the entorhinal cortex arrive to stratum lacunoso moleculare, while Schaffer collaterals input from CA3 pyramidal cells are the main source of projections to the rest of CA1 layers. In fact, further differences have been observed in the connectivity pattern of stratum lacunoso moleculare, such as a higher glutamatergic, serotoninergic, and cholinergic innervation. Due to this extensive connectivity, the hippocampal formation is implicated in a variety of functions, such as memory processes, spatial navigation, and emotional control, and there are many pathological conditions associated with alterations in the synaptic circuits of the hippocampus. Therefore, studying the exact way synapses organize in the brain is a crucial step toward understanding the functional organization of the brain. However, true synaptic contacts can only be observed using electron microscopy techniques. Since we don't know if the data obtained from animal models can be reliably extrapolated to humans, applying these techniques directly to the human brain is a major goal in neuroscience. In that sense, we have recently shown in our group that volume electron microscopy can be applied to the ultrastructural study of the human brain obtained from autopsies and biopsies, yielding excellent results. In fact, we already applied three-dimensional electron microscopy to study the synaptic organization of all layers of the CA1 field in the normal human hippocampus, and we observed that there were remarkable differences also in the synaptic organization of stratum lacunoso moleculare compared to the rest of CA1 layers. One of these diseases that is commonly associated with alterations in the synaptic circuits of the hippocampus is Alzheimer's disease, which is a neurodegenerative disease that constitutes the most common cause of dementia. There are 2 main histopathological hallmarks commonly associated with this disease, which are the accumulation of senile plaques and neurofibrillary tangles in the brain. In fact, the CA1 field has been observed to be especially vulnerable to both of these pathologies. However, differences have been observed between the layers, with the stratum pyramidale and stratum radiatum being the layers more affected by both of these pathologies, and stratum lacunoso moleculare being only mildly affected by the neurofibrillary pathology and remaining relatively less affected by amyloid pathology. Synaptic loss has been reported to be the best correlate to cognitive performance in Alzheimer's disease. However, it has mostly been inferred using light microscopy or conventional electron microscopy. Nevertheless, in a previous study from our group, we already applied three-dimensional electron microscopy to study the synaptic organization of stratum pyramidale and stratum radiatum of the CA1 field in Alzheimer's disease, and we observed differences in some synaptic parameters between the groups. However, since stratum lacunoso moleculare exerts so many peculiarities in its synaptic organization and its connectivity pattern, we now wonder if there may be specific synaptic alterations occurring in this layer in Alzheimer's disease individuals. So, to address this question, we use samples from the hippocampus from 5 subjects with no recorded neurological alterations, considered to be the control group, and 5 subjects with a different degree of Alzheimer's disease pathology. Samples were processed with different histochemistry techniques, and these sections were used for proper layer delimitation and for hippocampal atrophy and histopathological assessment. Additionally, samples were processed for electron microscopy to conduct an ultrastructural study of the samples. Specifically, we use focus ion beam scanning electron microscopy for this purpose. In this microscope, the focus ion beam mills the sample by scanning parallel to the surface of the cutting plane, while the scanning electron microscopy images this exposed surface. This process is repeated in an automatic and sequential manner, allowing the acquisition of long series of images. Applying this methodology, we obtain a total of 30 stacks of images of the neuropil of stratum lacunosum moleculare from both control and Alzheimer's disease cases. Synapses in these stacks of images were analyzed using SPINA software, which is a specialized software that permits the three-dimensional visualization and semi-automatic segmentation of synapses. SPINA software provides quantitative data from its synapse, and this data is known to have a functional correlation and can be used for the implementation of computational models. Additionally, the software allows the application of a three-dimensional unbiased counting frame to avoid the analysis of uncompleted synapses and permitting the exact calculation of synaptic density values. Since synapses are fully reconstructed with the software, they can be classified attending to the thickness of the postsynaptic density into asymmetric or excitatory synapses when they have a thick postsynaptic density, or as symmetric or inhibitory synapses when they have a thin postsynaptic density. Synapses are also classified attending to the postsynaptic targets into axospinous or axodendritic synapses. Axospinous synapses are further classified as synapses on the head or the neck of the spine, while axodendritic synapses are further classified as synapses on spiny shafts, when dendritic spines can be observed emerging from the shaft, or as synapses on aspiny shafts. Synapses are also classified attending to the shape of the synaptic junction, and size-related synaptic parameters are obtained from each synapse by extracting the synaptic apposition surface, which is the surface of apposition between the presynaptic zone and the postsynaptic zone. These size-related synaptic parameters are extracted with the SPINA software. So, applying this methodology, we observed that there was a 30% hippocampal atrophy in Alzheimer's disease cases. We also observed a variable degree of amyloid and tau pathology in Alzheimer's disease cases, while these histopathological alterations were absent in control cases, and this was as expected by the neuropathological diagnosis. For the ultrastructural study of the synaptic organization of the neuropil, more than 5,000 synapses were completely reconstructed and analyzed. Regarding synaptic density parameters, we did not observe differences in the density of excitatory synapses nor in total synaptic density between the groups. Nevertheless, a great variability could be observed between Alzheimer's disease cases in these parameters. On the contrary, a significant reduction in the density of inhibitory synapses was observed in Alzheimer's disease cases. In addition, we also found a lower proportion of inhibitory synapses in Alzheimer's disease cases when analyzing the excitatory-inhibitory synapses ratio, and this lower proportion was observed in both axospinous and axodendritic inhibitory synapses. So if we take all this data together, it seems to indicate that there is a specific loss of inhibitory synapses both in dendritic spines and dendritic shafts in Alzheimer's disease cases. If we now focus exclusively on axodendritic synapses, we observed in Alzheimer's disease cases a lower proportion of both excitatory and inhibitory synapses on aspiny shafts. These aspiny shafts are mostly originated from injured neurons, so this kind of neuron seems to be especially affected in this layer in Alzheimer's disease. I would like to remark at this point that none of these alterations in inhibitory synapses were observed in our previous study, neither in stratum pyramidale nor in stratum radiatum. So in this regard, this finding may have functional implications since GABAergic interneurons located in stratum lacunosum moleculare are known to form an inhibitory local circuit whose activity is sufficient enough to control the activation of CA1 pyramidal cells. Regarding the different synaptic shapes, we observed a higher proportion of excitatory synapses with macular shapes and a lower proportion of excitatory synapses with more complex shapes. So these findings, this synaptic alteration may also have a functional implication since perforated synapses are known to have more AMPA and NMDA receptors, and are thought to constitute a relatively powerful population of synapses with long-lasting memory-related functionality. Regarding the ultrastructural study of size-related synaptic parameters, although we did not observe differences in the mean area and perimeter synaptic values between the groups, we did observe that excitatory synapses with bigger area values were less frequent in Alzheimer's disease cases. So in that sense, the shape and the size of the synaptic junction have been strongly correlated to many synaptic efficacy parameters. If we now focus on the curvature of the synapses, we found that excitatory synapses were more curved in Alzheimer's disease cases than in control cases. And although the mechanisms are less understood, synaptic curvature has also been associated to synaptic efficacy. So taking all this data together, it seems that there is a reduction in total synaptic surface in Alzheimer's disease that is taking place through morphological alterations in excitatory synapses. This reduction may be underlying the impairment of synaptic functioning that may eventually lead to the learning and memory difficulties observed in Alzheimer's disease patients. So finally, in our study of the interindividual variability of Alzheimer's disease cases, we found correlations between the degree of amyloid and tau pathology and some synaptic parameters. However, extreme synaptic alterations and an extreme degree of tau pathology, such as bundles of thick electrodense material inside axons and dendrites, which are known to constitute the neurofibrillary tangles, were mostly observed in 2 Alzheimer's disease cases with an intermediate and advanced neuropathological stage. So to conclude, I would like to sum up these findings into the following take-home messages. There is a loss of inhibitory synapses both on dendritic spines and dendritic shafts in stratum lacunosum moleculare of the CE1 field in Alzheimer's disease, as spiny shafts, which are mainly originated from interneurons, have less synapses in Alzheimer's disease cases in stratum lacunosum moleculare, and since these alterations were not observed in other CE1 layers, it seems that interneurons are especially effective in stratum lacunosum moleculare under Alzheimer's disease pathology. Alterations in excitatory synapses in Alzheimer's disease occur through morphological changes affecting their shape, size, and curvature in this stratum. And altogether, the observed synaptic alterations may represent a structural basis for the progressive learning and memory dysfunctions observed in Alzheimer's disease patients. And thank you all for listening.